Learning objectives. I've listed a bunch of learning objectives here. We're going to talk about metagenomics. We've already kind of touched on that already. I'm going to give you a very high level of the idea of creating assemblies from raw reads, strategies for creating MAGs, as well as the importance of the quality control and suggested metrics for both assembly as well as metagenome assembled genome creation. I'm going to give you dip the toe a little bit with respect to Onvio. I'm going to show you a little bit about it, but most of the actual workshop lab is in Onvio today. So I'll show you a little bit about it, but you're going to get a good familiarity with that today. And then this is not the last thing that we're going to do, but during the talk, I hope that you're going to get a little bit of a feel for the advantages and the important caveats of MAGs with regards to, or as compared to assembly. So assembly and then the subset, which is creating the MAGs. What I really want you to actually come out of this with is that you'll never take another sample until you have a data management plan. So you should never collect another biological sample after this workshop until you have a plan for your data to do your data analysis. It is absolutely a crucial part of planning your research. If you make data that's not compatible with the analyses you want, you've wasted all of your money, all of your time, and that's bad. So let's not do that. Okay. So why are we using metagenomics? I'll just brush over this because Morgan talked about this, but we're looking to look at the complexities of microbial communities. We're very interested in their functionality as well as their evolution. We want to know how they're related to each other. We want to know how these communities interact with each other. So we're not just looking at single genes. We're not just looking at single organs. Taxonomic anchors such as the amplicon, the 16S amplicon, 18S, ITS, so on and so forth, are very, very helpful, but they do not give you a complete genomic view and a larger view of the complexity within a microbial community. That includes genomic plasticity, strain heterogeneity with respect to strains that are closely related to each other, transfer of mobile genetic units, or elements, excuse me, the plasmids that are found within certain types of microbes and other unicellular eukaryotes. It's important to note that metagenomics helps open up our ability to study organisms that are not able to be isolated and not able to be cultured. That's really the game changer for metagenomics, is that it allows us to access a part of the community that we are not currently able to do within the lab or bring into the lab readily. You can culture a lot of organisms, but it's very, very difficult, very time consuming. And we want to keep doing that, but we will always miss a large portion of the community. So metagenomics helps us deal with that issue. It also helps us look at the novelty within the community. And additionally, which we will be looking at a little bit tomorrow as well, is build out these metabolic models for both individual organisms all the way up to the community level metabolism. So we're going to start with cost because you're going to build your study throughout. We're going to essentially walk you through the strategies that you should be thinking about when you are building your study. Over the last few decades, genomic sequencing has gotten much less expensive. So this is a study from the NIH. I think it was published in 2023, uses data all the way up until 2022, just basically showing you that raw megabases of data has become cheaper and cheaper. The reason why I'm showing this, though, is that this is both a curse and a blessing. As we start to be able to produce more and more data, we have to manage that data. So in the Hallam Lab, we try to build our mind around a core concept of a data hierarchy, as well as this term, biological simulacra. So simulacra is a term that was made more recently made popular in the 1980s by Jean Baudrillard. He defined it as an incomplete representation, a proxy, a simulation, a distortion, a model of reality, or perhaps not a great model of reality. Every single thing that we create, every data product that we create in everything that we talked about today and earlier in the week and tomorrow, they're all simulacra, biological simulacra. They're models of what we have out in the environment, but they are not perfect. They have caveats. They can be very, very helpful, but we need to understand when we can use certain ones and how they can be used together. So the data hierarchy starts at the top with highly known and studied organisms, such as E. coli, which is arguably one of the most well-known microbes on the planet. We have lots and lots of information. It's highly curated. We have lots of research in lab, in silico, in the field for understanding E. coli. So it gets the T1 designation. There are very few T1s out there. Moderately curated organisms that are still isolated, perhaps, fit the T2 through T3, depending on how much information and how confident we are about that information. Largely, the environmental genomics type data that's available in databases are not going to be tier 1 or tier 2, maybe not even tier 3. So you have to be very cautious about how and when you're using this type of information. Lastly, the culture-independent methods, so this is what we're talking about today, metagenomics, sits at the bottom of the pyramid. That includes raw metagenomes, which is what we did in the first session today, and then MAGs, as well as I put single amplified genomes there as well, because they are those are culture independent, but they are slightly different than a MAG because they are isolated before they are sequenced, not after they are sequenced. So they're isolated in a tube, they're sequenced, and so we have a different assessment of the confidence of that data. If anybody want to talk more about SAGs, I do a fair amount of work with SAGs, but we're not going to really do a lot of that today. So I just wanted to throw it on there to so that we had all the parts available. So when you're planning out what type of study you're going to be doing and what kind of data that you're going to be producing, it's important to understand some of the properties of these different data. There's short read and long read. We can incorporate those together as hybrid with respect to the assembly, and I'll talk a little bit about that later. But when you're looking at the raw data that you're getting, and also planning out what type of data maybe that you might be able to have, you have to think about your DNA concentrations. You have to think about the quality of the DNA that's going into your sequencer. You can't get long reads from short input DNA. It's impossible. Okay, so there are limitations to these different types of platforms, and it's important to understand that. Short read is by far the most popular. Most of the data out there today is Illumina short read or some form of short read data. I put some stats on there with respect to read length and error. I'm not going to walk through every single thing, but just understand that each of these has a place. And as technologies improve, that's going to change. And so keeping track of that, and again, understanding the type of data and the quality of the data that you're going to be able to produce dictates what kind of platform you should be focusing on. Additionally, your budget also dictates that. So this table, again, it's a little bit small. We don't need to worry about it. You can look into it. I did this on my own, so it's not all going to be perfectly accurate, but it gets the point across that there are per sample, there are per run differences between these different technologies. Illumina is by far the cheapest with respect to high throughput, but long read is closing the gap with respect to the PacBio Hi-Fi. And especially, and I was talking to Stephen about this earlier, if you're looking at per base or you're looking at per run, perhaps there's a big difference. But if you're looking at per genome or per mag, you have the possibility of making very, very high quality population genomes from long read data. And so if your interest is to find these genomes, you may actually do better spending a little bit more per base pair on Hi-Fi if you have the possibility of having more genomes that are higher quality at the end of your process. Okay, so what is a metagenome assembly? A metagenome assembly is a way by which we take raw reads of any kind that we have, and we use a type of Kamer-based analysis to combine those. Yes?
And additional costs, the form costs, but there's also the cost of writing stuff in front of there.
Absolutely.
So if you comment on, whether the sequencing is really a real cost, relative to the driving in the that's more expensive.
So I can't speak as an authority, but what I will say just from my own experience, I actually find that the most expensive part of the sample is getting the sample, like going out and planning out and getting the actual biological sample. In most cases, it's not necessarily a money cost, but it's an incredible resource cost. But with respect to library preps are expensive, but they tend to be able to be scaled to a degree that is a little bit more affordable than the actual consumables of certain platforms. So as an example, PacBio uses smart cells. Those are a one type of unit that can hold a certain amount of information. So a certain amount of DNA. You have to buy the smallest version of that, and that's like an upfront exact cost. There's flow cells on MySeq and so on and so forth with respect to and PacBio or Oxford has its own as well. But I don't know, Morgan, do you have a sense of like what's the most expensive part of the process? Yeah. Yep. Yeah.
Why it's really good. A plateau or lower places, but
Yep. And I'll reiterate, data management plan. Those people cost an incredible amount of money. Sometimes you will not be able to get your sample. And if your samples from, let's say, so we collect samples occasionally from the middle of coastal estuaries or out in oxygen minimum zones. You have to get a boat. You have to go out there. Actually, I have some really cool pictures. It was a hovercraft. So, I'll show you those later. That's super cool, but that's not cheap. You could sequence hundreds of times over for the fuel cost to go out and collect those samples. So you want to make sure you have your plan. Now, there's always things that come up with respect to your data, but having a plan beforehand is as important as having the staff, as important as having the biological question. You have to have that plan. I'll keep saying it, because I have spent an enormous amount of time with other people's data that they didn't have a plan for, trying to wring out some type of statistical significance for them and feeling terrible when I can't, even though I didn't do it. Like it wasn't my fault. But so just be aware. I'm going to keep saying it. Okay, metagenomic sampling. It's a type of K-mer analysis. And it brings us back to Königsberg. So anybody who was here earlier in the week, I gave a little story about Königsberg. This is a story of the 7 bridges where the wonderful people of Königsberg wanted to figure out if they could walk across each bridge one time only to get from one place to another place. And so they charged Leonhard Euler to figure it out. He was a famous mathematician at the time. And I won't go way into it, but in doing this process, he started to lay the foundation for graph theory. So he abstracted the land masses and the bridges into a graph, which had nodes and edges. And then he determined that it was not possible. If you want to know more about why that is, there's a really nice mathematical proof that came out a little bit later that shows that if you have an odd number of edges going into a node, you cannot find a path through that goes through every single edge at once and only once. And so the bonus that's actually a very trivial thing. Once you've figured out that if all you have to do is count the edges or count the edges going into a node, you can do that really, really quickly. So that's extremely trivial. What's not trivial is if you're trying to determine the shortest path through each node once. That's called the traveling salesman, and that's actually a not solved problem. So there's one way by which it's completely trivial to figure out what the path is, and there's another way that it's basically impossible. It's not impossible, but if you try to brute force it, it's a factorial, which means every time you add a node, you multiply it by the number of nodes in there. And so it becomes infeasible to compute. There are heuristics to do so, and lots of different graph-based analyses use heuristics, but then they also leverage the fact that Euler figured out the trivial way to do it. So I'm bringing this up because metagenome assembly is a K-mer-based network analysis or graph analysis. So instead of what we did earlier in the week, we did correlation analysis where the nodes were ASVs and there was a correlation number that was associated with each edge. Now we're building a graph that uses k-mers. So on the left here, this is like little sequence, and we break it into, in this case, 4 mers, so 4 base pair k-mers. Each of the nodes is a k-1 string, and each edge is the actual k-mer. And so what you're trying to do is you're trying to find if there is a path through your network. And so by using Eulerian circuits and mathematics, we can find the path through the network. Now, it is trivial to do it for 4 nodes. It is trivial to do it for a couple more nodes, but it isn't necessarily trivial to do it for thousands and thousands or hundreds of millions of nodes. And so that's why we actually do still need software and still need heuristics to be able to do this process. But it does make it feasible. So what we do is we create our network, we find our path. That means we take our reads and we stitch those reads together to create more contiguous segments. That's why it's called contigs. So contig stands for contiguous sequence or contiguous region. And then afterwards, if we have paired end information, so paired end reads forward and reverse, there are occasions when the graph will break and we'll have segments that don't overlap with each other. But if if the forward read and the reverse read are found on the 2 separate parts of these 2 contigs, we can associate them with each other. And so that's called scaffolding. And that's actually very important because it gives us more better contextual information with respect to the longer contigs. Okay, so we talked a little bit about assembly. Now we want to talk about producing mags from that assembly. So a mag is a population level simulacra. What I mean by that is that it's not a strain, as much as it's been reported in certain publications that it is. It's not. They are a collection of, think of it like a cloud associated around a region, and we call that cloud a population. So it can include, sometimes it can include not a lot of variation, depending on what kind of organism it is, but it absolutely can contain variation with respect to the strain heterogeneity of that genome. So it's a really nice organizational framework if we're interested in breaking the community into components and starting to get a feel for what populations may be responsible for different types of metabolism, whether it be contributing to the same metabolism or collecting the outputs of one metabolism to do another metabolism. This happens a lot with respect to anaerobic environments that I work in. So the denitrification pathway that goes from nitrate all the way down to dinitrogen gas is separated into different parts that different organisms do at different points in time as well as in space. So that's a really neat thing to be able to do. And in order to do that, we need some type of organizational framework. It lets us further extend the metagenomic information that or annotations that we have already done. So you can start to associate those individual annotations that you got from, say, Kraken with each other to then start to build an idea of what these populations are capable of, potentially. And if you want, you could do metatranscriptomics and then show what they are actually trying to do. So that's a very, very important. So it's essentially the T4, so that's metagenome with more structure. And that's really, really neat, but there's a catch. And the catch is you have to account for your data loss. So this is a real figure, real data. It's not been published, but it is real. And this is showing from left to right, raw sequence data in reads, all the way to high quality mags and the loss that is seen when you do this. Now, I would like to say that these are totally crappy samples and you're never going to see this. This is, generally speaking, this is what you're going to see. Mags are going to make up a very small amount of your actual metagenomic data. That doesn't mean it's not important. It doesn't mean you can't say something about it, but you have to take that into consideration when you are talking about mags. It is a subset, and generally it is a very small subset. Again, you can say really, really important things about a community with that, but it's important. And this is why I spend a lot of my time trying to integrate different omics data being able to contextualize your mags within your assembly and your assembly within your environment and so on and so forth is very important. Go ahead.
Like, or in that example, then you're gonna get more mag.
Maybe, I was saying maybe. It really does depend on your environment. Often the limitations to getting quality mags is it's more complicated than just sequencing more. Absolutely, if you sequence more, you have a higher probability. However, if your community is extremely diverse, you may not be able to sequence enough to get that. You're going to continue to get the very common things, and you're going to continue to make more of those mags. But if and this is the case for most environments, most organisms are rare. Not all the time. Maybe they're situationally rare, but they're rare. And so it can be quite a challenge to get enough coverage. Now, that doesn't mean that you can't, but that's something to take into consideration. Additionally, if you have a lot of strain heterogeneity, and I don't think I define that term, but I will define it now in case anybody's wondering. Strain heterogeneity is essentially a measure of if you have organisms that are closely related to each other, let's say they're in the same species or even the same strain, they're going to have a lot of variation with respect to their genome, or they can. And if that variation is very high, binners struggle terribly with that. They do not do a good job with that. And they don't do a good job with a lot of things. That doesn't mean they don't do a good job with some things. But again, these are all things that you have to take into consideration. I'm not really selling the whole mag thing, am I? No, they are really, really helpful, but.
Like, my examples have low minutes. Is it, like, would it be silly to even try?
It's never silly with the understanding that you may not get anything good, and you definitely need to take into consideration all the things I'm talking about. So the less data you have, the less probability you have of getting complete information. You may be able to get contigs. You may even get a few mags, but you have to be very cautious. And this is not just with low biomass samples. You have to be very cautious with how you quality control and how you interpret. And part of what we're doing today is taking some mags and refining them. So that's part of the process that we're going to be looking at too. Yeah. Okay, so pros and cons of whole genome versus mag. I talked a little bit about these things. The metagenome gets a lot of DNA, not all of it, but a lot of it. The mags are in some cases a very small subset of that. There are biases to binning, as I said, as I showed with the data loss. You will lose data, and the thing that you're interested in might not be in those bins, and it's very likely that it won't. I'm not going to read through every single one of these, but just it's very important to weigh these options. You can always run an analysis. That's generally fairly painless, but if you put in something that you don't understand, you're going to get something out that you can't understand or that you can't interpret. And so that's very important. Okay, now how do you make a bin? Let's talk about that. So largely, most binners do pretty much the same thing. Now, that's not entirely true. They use all kinds of different algorithms, but the inputs to the binning algorithms are largely the same. The binning process is essentially just a clustering algorithm that takes a feature table. And the feature table is again, this is largely common, to tetranucleotide frequency. That means a former, you actually count the relative abundance of all the different tetranucleotides on your contigs, and then you can cluster your data based on that. Additionally, you can use abundance information. And this is very important, particularly for determining the difference between closely related strains that may have very different abundance profiles. So think of it as a clustering algorithm that the input is the tetranucleotide frequency, similar to an ASV relative abundance table, but for the k-mers, and then the abundance of each of those contigs within the different samples. And you'll see that today with what you're analyzing. Okay, so there's tons and tons of decisions to make. There's budget, there's different types of sequencing platforms, there's all these different types of software that I have told you nothing about because it gets really confusing really quickly. But CAMI has helped us. So CAMI stands for the Critical Assessment of Metagenome Interpretation. And they are a group, not the only group, but a good group that has started really trying to get at benchmarking and comparing bioinformatics software, specifically for metagenomics. So when you have questions of which assembler should I use for my environment, you can start with some of the publications and some of the data that CAMI has provided. And I'll show you not the data, but I'll show you some of the data, but I'll also tell you where you can find that data as well, and those in the publication. There's currently 2 main publications, CAMI 1 and CAMI 2. What they do is they run these challenges. Anybody who has a bioinformatics software can download their challenge data set, run their software on that, whether it be an assembler, whether it be a binner, whether it be a taxonomic profiler, and then they collect the output of that, and then they score and compare all of those different software. And then we get the benefit from seeing how those different software do. And the answer, or the answer to that, is that they vary quite a lot. So the initiative is really, really cool. They have these benchmark data sets. They have a set of different metrics that they provide, and then they also calculate. And then they talk to the community, and they have a blog, and they have a website. So the website is right there, CAMI Challenge, or cami-challenge.org. And they really, really do help you make an informed decision. So this is a plot. These are all quite small, and so I don't expect you to interrogate them deeply, but essentially this is the kind of plot you get, where each of these facets is a different metagenomic assembler. And what we're looking at is we're actually looking at a curve that's plotted for how much coverage do you need for your metagenome for each of these assemblers. Now, what they use, and I should be clear that these benchmarking data sets are based on, they're fully tractable reads. So they keep track of the reads from the absolute beginning of the process all the way to the end of the process, which allows us to know the actual truth of where these reads should be. And that lets us assess these software in a way that we really didn't get to do before. And so this has to do with sequencing coverage, because a very common question is, how much sequencing do I actually need to do for my assembler to work on my environment? And so this can help you with that. There we go. These are radar plots that essentially are showing different metrics for assemblers. And you can see that not every single color here, which is a different assembler, they're not all maximal at different with respect to everything. And so there are different assemblers for different jobs. And that can get a little bit daunting, but this is starting to get at that issue. Click. There we go. And then lastly, this is a plot that shows the assessment of binning. So each of these bars, each of these colors is a different binner. And these are just metrics that are showing things that we will be looking at today: completeness, contamination, strain heterogeneity. They can calculate these purity in this case, which is the inverse of contamination. They can calculate different indexes on this, and it's not an estimate in this case. Many of the tools that we use are estimating it based on a set of marker genes or things like that. These are actual numbers because they know exactly where every single read goes. So that's really, really helpful that we don't have to estimate in this particular case. Okay, so in this year, CAMI released a portal on their website. So now there's a way to actually go through their data and look through little plots. I think it's a work in progress still, but it's definitely a very, very nice thing to have. So I would suggest everyone take a look at that. And there's a ton of different publications and actually most binners that are contemporary have some type of information that they've used in CAMI. I personally built myself a binner and I use CAMI for it. So it's, again, a really, really nice thing to be able to do. Okay, short answer, CAMI helps, but there's no best. And that's because it's more complicated than just saying that there's one metric. There's many, many metrics. There's many, many decisions made. All the things I said before with respect to budget and available resources, so on and so forth. But with respect to the software, it actually turns out that many studies have shown that an ensemble approach is actually ideal. So that an ensemble is when you take multiple softwares, you run them, and then you combine those. And there's all kinds of different ways that people combine them, but the idea is that you're essentially averaging out the positive and negative qualities of those assemblers or those binners to get a better result than you would get individually. And so we're not doing that today, But when you're going to get the opportunity to use a couple different binners today, and you're going to see that they give you different results. And that's very, very important to understand. How am I doing on time? Am I good? Yeah. Okay. Yeah.
So, I mean, there's a lot of different efforts so forth. You do one. What do you?
I don't do one. I sometimes do one depending on the data. So as an example, there are a number of different metagenomic assemblies. MegaHit is a very, very common one, and it does a very good job. It is also extremely resource lean. It doesn't use a lot of RAM. You can give it 1 million threads and it works just great. It's awesome. However, it isn't actually the best, depending on what you're looking at. You can often get a better assembly from Metaspades, but Metaspades is very hungry. It uses an incredible amount of resources and time. And so I tend to use MegaHit because it is the balance of it is good enough and very good enough, and it does not eat up all the carbon on the planet. It doesn't burn all of our resources. If I am struggling with trying to get an assembly to work, I'll jump to Metaspades. And there are tools out there that will allow you to combine those. I'm blanking on the MetaWrap. MetaWrap is a pipeline that actually it's more like an ecosystem, but it allows you to combine a Metaspades and a MegaHit. I don't find that does a lot for me, so I don't generally do it. Now, with respect to binning, I do not ever single bin. I always use an ensemble. That doesn't mean that I don't get good bins from singles, but I just don't. Binning is actually not that expensive computationally. So I use an ensemble approach. I do a bunch of different binners and I combine those somehow. And I've also built one of my own binners that I haven't published yet, so we don't need to get into it, but if anybody wants to know about it, it's pretty cool. And it also allows for an ensemble approach that if I give it, I can also give it bins from other and then use those as kind of like almost like an anchoring. So, yeah.
My impression is that there's an assumption that MetaRab, it gives you, say, each combination of prediction.
Mm-hmm.
So you have method A, method B, and method C, and then in that prediction, just you can know just you can see. Then the one's combined A and B, one's A and C, B and C, and A, B, C.
Yes.
And so I think there's an assumption that people would assume that the A, B, C, that combines all three predictions is best.
Mm-hmm.
But that's not necessarily the case, is it?
No, it's not.
Yeah.
Yeah, that's right. So we don't have to get into the math or the algorithm behind each of the binners, largely because I won't be able to do that, but it's not always best to combine. And a great case of that is if you have a very high strain heterogeneity, binners struggle terribly with that. And so if you combine multiple bins that are fairly low quality, you just get a bigger bin that's even more low quality. And so that's not a good strategy. However, you can absolutely get better bins with that protocol or that strategy. And when we're doing right at the end, the bin refinement, you'll remember this, when we're doing bin refinement at the end of the actual lab, you'll see a circular plot, and I'll show you what those look like soon. And it will show you how you can actually refine a bin based on the coverage that you see. And it's surprising how much variability a binner actually will think is the same organism. And that really has to do with strain heterogeneity, at least in the case of today. But yeah, so I don't need to keep rambling about it, but it's definitely, you don't just combine everything and say that's better than individual. Actually, a great example of this is, in this balance between performance and with respect to quality, the JGI, the Joint Genome Institute, which is in the United States, it's a big sequencing center that also does a ton of bioinformatics, and they put out really high profile metagenomic based studies. Their pipeline is they just use MetaBat2, which is a binner, which you'll hear about today, because it is for a single binner, it averages better than most. Not in all cases, but in enough cases that they chose that they would just use one. So there's all kinds of different reasons that choices are made. If you're at scale, maybe you can't ensemble, but we can all do ensemble, and we definitely all should. Okay, so there are groups out there that are trying to stabilize and stabilize the quality metrics, set out a list of quality metrics and guidelines for quality of assemblies, quality of mags. I'm not going to read the whole table, but these are the metrics these are some of the metrics, and there's a publication that's associated with this, actually a number of publications. It's Bowers et al. That was kind of like the first, it's the MIM-SAG, MIM-MAG. So that's minimum standards for genomes. So it considers the quality of the assembly. It considers contig number and how contiguous those contigs are. If you have marker genes such as RNA, if you have other things such as other genes such as tRNA, what kind of taxonomic classification, so on and so forth. Nobody makes you do this stuff. You just have to feel like it's right. So, and I want you to feel that it's right. Keeping track of this, the quality of these things is very, very important. Another kind of not necessarily a group, but a strategy that's being employed and guidelines that are being laid out is the findable, accessible, interoperable, and reusable. The FAIR principles. This is for data management. This means that you want your data to be findable. You want your data to be accessible. You want your data to be you want your data to be understandable and your metadata to be understandable, and you need it to be reusable. These are, again, no one's making anybody do this, but you should follow these rules, because if you want your science to be reproducible, you have to follow these guidelines or something very nearly these guidelines. Yes.
Some funding bodies require you to have that.
It's true, yeah.
And somebody actually does.
That's great. No, if you're required to do a data management plan for government proposals and things like that, you're absolutely right. Many of them require this now because too much money has been wasted on making data that then can never be found again. So, okay, so that's great for your data, but what about your process? How do you keep track of your process? And so the way that you do that is through workflows. Something as simple as the things that we're doing in each of our labs. You can consider these workflows. We're walking through, we have documentation, we have environments, and we have the actual commands. We have the data. So that can be considered a workflow. There's lots of different ways to version control. There's lots of different ways to have workflows. This is a glyph representation of a workflow. We use these in the Hallam Lab a lot to describe our process before we do it. This has to do with wet lab. This has to do with dry lab. It has to do with the integration of wet lab and dry lab. So in this particular case, what I'm showing you is an amplicon and omics workflow that I use commonly for presentations that shows you what I happen to be doing or what we happen to be doing. In this case, the orange nodes and edges are what we're doing today. So you can walk through this process and you can see the flow of this process. This is very high level, but what it does allow you to do is get a feel for what components you have in place, what components are still experimental, what components you need to find someone to help you with. And someone can very easily look at this from me and say, I see what he did with his process. You can then overlay this using something like a workflow management system, like Nextflow or SnakeMake, onto high compute systems. Don't worry about all these icons. Don't worry about it. I just wanted to show you that you can integrate this into high performance computing, such as like what we're doing on the cloud today and during this workshop, using a number of different of software and services. And that allows you to then further layer onto the reproducibility and the structure essentially of your workflow. So this is actually a visualization of our infrastructure in the Hallam Lab. We have our personal computers that everyone has either in their lab or with them, their laptop. We have cloud compute. We have heterogeneous compute cluster. We have cloud storage, and we have a way by which all of that data can be transferred on and off of all of those systems. I created this modular architecture for data management and analysis when there was actually a cyber attack on UBC, which shut down all of our infrastructure at UBC. And so it just so happened we had an allocation we weren't using for the cloud. And so during that shutdown, we needed to get our work done. And what I did was I built out the cloud infrastructure. I plugged it into all of the different components, and then our lab was able to continue doing data. Now we had to do it because it was something bad, but it ended up being something far greater than we thought, because it now has grown into something where we can store all of our software, all of our data, all in one place. Everyone can access it from wherever they are. And it helps all of our teams and our collaborators work very efficiently. This is the future, maybe not this particular thing, but the cloud experience that you're getting today and during the workshop is extremely valuable. So consider that. Okay, omic specific analysis. So we're going to be looking at Onvio today. Onvio is an environment that was created by Marin, who is, I think he's currently in Germany. These are just quotes from their actual website. Through modular design principle, Onvio resembles Lego, where the same simple blocks can build non-identical complex structures. So this graph that you're looking at is actually the ecosystem of Onvio. Each one of the nodes is a data type or a type of analysis, and the edges connect the ones that are compatible with each other. And so what you can do is you can literally go onto their website and look at this graph, and you can follow it through to the analyses that you want to do. And they have an incredible amount of documentation. And I actually do want to just take you on a quick tour, if that's okay. Not super fast, but we're just going to take a quick look. So this is the website. The people, they're all about reproducibility. They're all about attribution. This is Marin here. They have a Discord. It's installable on all kinds of different things. It gives you it's very, very polished as well. There's a community blog, but the most important thing that I think, that other than actually knowing how to install it, is I want you to remember this. So they do amazing tutorials. Anything you want to do with respect to genomics, not anything. Marin would be the first one to actually tell you that Onvio is not great for everything. It's just great at a couple things. And what it is, I personally think that what it is great at, it's really great at. So they have tutorials here, Introduction to Unix, they have Metagenomic, they have SAGs, they have functional analysis, so on and so forth. They have workshop materials. They run amazing workshops. I've had the privilege of working with them in the past. They are incredible. They have technical write-ups. They're all about open source, open documentation, good quality documentation. So if you ever want to learn more about it, you can go to this website and you can actually learn all of it here. You can join their Discord and talk to them if you want to. They're very friendly, actually. And they do a really good job of enabling people to be creative with their science and creative with the discovery or their discoveries in their data. So anyway, I'll stop gushing and we'll go back to the presentation. Okay, so Onvio is cool. We're going to be doing that today. This is a figure that you're going to be creating. Not exactly these figures, but something very similar to these figures. Maybe exactly, depending on what journey you take today during the lab. So what these are visualization of a genome, of a MAG, actually. Well, actually, we should call it a bin first because we haven't refined it. Then you call it a MAG after you refined it. On the left side is a bin that's considered 100% complete based on marker genes, and it has a low redundancy. This term redundancy is something that the Onvio crew use, and I do agree with it, instead of contamination, because it's not contamination until you've identified it as contamination. Just like we were talking about non-target versus host versus true contamination. Unless you've actually identified it as contamination, it is not. It's just redundancy. And many genomes have redundancy of single copy genes that are actually real. And so you do have to be cautious about that. So this one on the end, on the left side, you can see that each of these little bars is the abundance of the contigs. The dendrogram in the middle, each of those is a split of a contig, and then it's a clustering in the middle, and then each of the little bars is a sample and the abundance of the sample. So the more black you see across a bar, that means it's a higher abundance. And the immediate thing that I want to draw your attention to is the completeness and redundancy of the left and the completeness and the redundancy of the right. These are the same species. Look at my air quotes, same species, right? But you can see that the abundance profile of the right one is very, very different and very inconsistent. And that is indicative of something that may have a fair amount of strain heterogeneity. There may be more than one genome in there. They may be half of one genome and half of another. What you're going to do today is use the tools that that Onvio has and be able to highlight different sections and refine your bin into something. Now, I won't give it away and tell you what you get when you do this, but if you follow the protocol, you'll be able to see what you actually get when you refine it. It's a population genome. Yeah, so this is a this is a bin, and it's population genome. And so both of these are population genomes. One is parts of multiple, maybe strains, perhaps. They are both being identified as the same microbial species. And so the likelihood is that there's multiple strains that have different abundance profiles depending on what sample they're in or where they're. Each of these would be considered a single mag, made by two different processes. The one on the left was made by Concoct, which is a binner, a type of binner, and then MaxBin 2 is another strategy of binning. You're gonna get the opportunity to use both of those. And so just because MaxBin made this conglomerate, don't throw shade at MaxBin. Sometimes it does a really good job, and sometimes Concoct does a really terrible job. This is what you have to be prepared for, is you should never just publish bins. If you're gonna publish a bin, you need to look at it and refine it. There's far too many bins out there in these huge collections, and so many of them are crap. It's really a shame because it misleads our ability. Like, we can't download that data and then use it. We have to go through all this rigmarole to make sure that it's quality. Go ahead, sorry. The exact same assembly. Each of these profiles are a set of all of these are samples from the same study. Two different algorithms, clustering it two different ways. That's right. And notably all the other parameters are the same, other than the binner. And if you added another binner, you get another. And if you added another binner, you get another, so on and so forth. Okay, so that's pretty much it. If anybody has any questions, yeah, go ahead, Tony.